Everybody. My name is Roz Anderson. I'm with the Division of Geriatrics and Gerontology in the Department of Medicine here at the UW School of Medicine and Public Health. Hopefully you all are as excited to be here as I am. I think we've got a great program. It's my honor to introduce the first set of speakers in our first session, Epidemiology and Demography of Aging and Dementia. And so to keep things moving along smoothly, I'm going to have my timer. I'll be watching carefully. You'll see indications as I wave at you if you go over time. Our first speaker is Luigi Ferrucci, who visits us from NIA. Luigi was trained in Florence, Italy, and I know we have an Italian contingent already in the room. No waves. And so his role is as Chief of Longitudinal Studies at NIA, and he's also the Scientific Director of the Intramural Program at NIA. You have a nice little bio book. I encourage you to read that to see how accomplished and superb our speakers are. Let me just say that all of the longitudinal studies ongoing across the globe have had some contribution or other from our next speaker, who's really done tremendous work at allowing us to understand the biology of human aging. Please come.
Thank you for the nice introduction, and thank you, Sanjay, for inviting me to this meeting. When you asked me to come and talk in this meeting, I say yes, because I know that everything you do is really good. And I never imagined that there would be such an incredible assembling of speakers and program. So I'm really honored to be here and humble because of the level of all the other speakers that will be talking after me. Let me see if I can get my slides here. Is somebody going to help me doing that? I just need to... Okay, great, yes. Okay, so what I'm going to try to share with you today, within the constraint of the time that I'm allowed, is a glimpse to the future. You know, we have done a number of epidemiological studies, and I've been involved in many of them, and I'm enthusiastically work on delineating the trajectory of every single physiological and biological function as they evolve with aging. And the model that we have created over the year and we follow can be summarized in this very simple slide, where, you know, there are a number of domains, and these are the domains that change with aging. Everybody change in body composition, in energetics, in homeostasis mechanism, and neural control and neuroplasticity. This is not a disease. This is going to happen on everybody. There is no exception across organisms as well as in humans. And so that was really the main purpose of my research over the last 30 years, and understanding, you know, what are the risk factors, the consequence of these changes. But over the year, we have, I wouldn't say exhausted this part, but I got somewhat not completely satisfied by simple descriptive research, because I wanted to know why. You know, I was the kind of boy that was breaking the toys all the time because wanted to know what was in it. And at the same time, you know, when I started doing epidemiology, I wasn't really happy to know how change in body composition occurred with aging, but I wanted to start to know why. And the reason why we need to know why is that we need to intervene on them. In order to understand how we can intervene on them and find new target, understanding mechanism is absolutely fundamental. So after many years, I started thinking that the pond I was swimming in was very comfortable, I was very successful, but was no longer satisfactory. So I jumped out in a much smaller pond that I was not completely comfortable with and studying the biology of aging. What are the basic mechanisms that may be underlying this change that occurred in the phenotype? And these I delineated only, those that were, outlined by Lopez-Otin as possible change that occur and that are driving some of the change in our physiology and anatomy that occur during the aging process. I'm certainly not going to talk about all of them, but I'm going to drive you through an example to show you how even working in humans, even working not in animal model, we can start trying to apply some of the techniques and the technology that has been developed in animal studies to start understanding mechanisms in humans. So the domain that I'm going to target today is mostly body composition. And if I have time, at the end, I'm going to also tell you about a new study that is started recently at NIA that is called Gestalt, that is preparing for the new generation of longitudinal study of aging. Okay, this is a very old slide. Jeff Metter was at that time at NIA, is no longer there, and show a very simple concept. When you look longitudinal cohort, here you see that when you look at knee extension torque, which is a measure of torque that takes into account the leverage of measure and the muscle cross-sectional area, the mid-thigh calculated with CT scan, you can see that there is a clear decline of both those parameters, but there is an excess decline in muscle strength compared to muscle mass. And that means that not only the muscle will shrink and become lower as part of body composition, but their ability to generate force is much reduced. And you say, this is a curiosity. It's much more than a curiosity, because if you think about that, the only thing that we have done pharmacologically to try to resolve sarcopenia is increasing muscle mass. And increasing muscle mass clearly is not a problem here. The muscle mass decline, but very little. What we really need to do is increasing muscle strength. And so the billions of dollars that we have spent by developing molecules to improve muscle mass were really not well directed. In fact, most of those studies have shown very disappointing results. So what can we do to understand muscle quality? Well, one of the things that is pretty apparent is that the composition of the muscle really change with aging. And here I'm just showing you from our Gestalt study a series of MRI of the thigh that I ordered by age. You can see that, yes, there is an increase in fat infiltration and marmorization of the muscle, but it's really not the entire story. This man, who is 83 years old, came to our study and tell us that he needed to go home in the morning because he swim 2 miles every day in the lake, every days of the year, including during the winter. And of course, when you look at the muscle of this guy, you can see that there is a little bit of difference with the 26 years old, but not very much indeed. And even the spirit, I will tell you, was 14, not 26. And so there is some behavioral changes and some effect that counteract the effect of aging for sure. So how is happening? What is this happening there? So to understand that, the first thing we did was say, well, there may be a metabolic signature. There may be some metabolic parameter that change with aging. And the first thing we did, because we can't really go direct in the muscle, we use a signal in the blood. And we did the metabolomic studies nested in the Baltimore Longitudinal Study of Aging. The study was very simple. We look at muscle quality that was operationalized here as the ratio between strength and mass. And then we saw that, on general, muscle quality decline with aging, but there is a very large heterogeneity in the population. So we selected pairs of individuals that had the same age and the same sex, but had very, very different muscle quality. And we did a shotgun metabolomic study to see what was different between these two groups. And it was very interesting to see that what was very different is summarized here. So on the right, you have the metabolites that in the plasma were overrepresented in people with low muscle quality, and on the left, they were underrepresented in people with low muscle quality. And you can see immediately that those that were overrepresented in people with low muscle quality are amino acids. All the red there are amino acids. And especially high and statistically significant were the tryptophan and the long brain chain amino acid, which are really, really important to stimulate catabolism. But you will expect anabolism, but you will expect the opposite. You will expect that people with the problem in muscle quality will have low amino acids. And so this was a really positive results. And so what we did was to start doing biopsies. And when we did biopsy, things became a little bit more clear, because if you look at these three brain chain amino acids and the total brain chain amino acid, you can see that in the muscle, the proportion is absolutely opposite. So you don't see higher amino acids, but lower amino acids. So the problem here is that the amino acid cannot really enter into the muscle and remain in the circulation. The uptake of amino acid is reduced. This is really important, and I'm not going to spend too much time on this slide, just to say that the uptake of amino acid occurred through these two channels, and when we look at proteomics, these two channels were really not reduced with the reduction of muscle quality. But and when the brain chain amino acids don't enter into the muscle cells, they cannot stimulate mitochondrial biogenesis, and also the brain chain amino acid catabolism is essential for the creation of some of the metabolites for the TCA cycle, which is essential for the energy within the muscle. And also they stimulate mTOR, and through mTOR interact with the protein synthesis. So we have a problem in the protein synthesis, we have the problem with the mitochondria, and we needed to study that. So we thought there may be energetic issue, there may be metabolic issue, there may be structural issue that occur within the muscles. Okay. So in order to look at the mitochondria, we needed to use a technology that is called P31 spectroscopy. It's an MRI technique. It's based on the fact that in the muscle and in the brain, you need stable ATP, whatever the condition is, because if a lion is coming to you, you have to escape. You don't really have the time for your mitochondria to start generating energy and activate your muscle. And so the nature have selected this mechanism by which the energy is accumulated in phosphocreatinine, and then you use the energy that is in phosphocreatinine to feed your muscle, and when you rest, you recharge the energy of the phosphocreatinine. So what we do is that we exercise people in a magnet, and then we stop exercising them, and we use a special probe that can measure phosphocreatinine and inorganic phosphorus. And so this is the typical spectra where we see this is inorganic phosphorus, this is phosphocreatinine, these are the three species of ATP. And so during the exercise, the phosphocreatinine is depleted, goes down. And then when you stop exercise, the mitochondria start, mighty producing energy so that they can recharge the phosphocreatinine. And how fast they can recreate the phosphocreatinine is an index of mitochondrial function. So we can measure mitochondrial function in vivo, and we're done in VLSA at this point in more than 400 people, which is the largest population ever studied with this method in the world. And when we look at that, to make a long story short, we found that the parameter that express this mitochondrial activity, which is a KPCI, is a constant that integrates from the recovery curve, was strongly associated with mobility, walking speed. And the association was much larger for fast-paced 400 meters, which require endurance, but was a little bit lower for those activities that do not require endurance and just are important for rapid contraction. And then what we did, we said, well, what is this mechanism? Why is mitochondria associated with mitochondrial function? We expanded the population, and this paper is published now in Aging Cells, showing that in a mediation analysis, the effect of mitochondrial function on walking speed was mediated by muscle characteristics and muscle stress. So there's a problem in mitochondria, there's a problem in muscle, and this is affecting the decline of mobility in older people. But also, as you remember, we said that maybe structural problem. In order to look at the structural problem, we started using the technique that's called diffusion analysis. Now, we have seen, all of you have seen beautiful images of diffusion analysis from the brain to look at, the white matter connections. And we apply this technology to muscles. It was a little bit tricky because there is a lot of fat interfering, so we had to work on a fat suppression algorithm. But in the end, we were able to do that. And when you do that, you get really interesting images. The images you get are like this that look at a parameter that is called anisotropy. Let me get back a moment to you to tell you what anisotropy is. Anisotropy is the ability of the water to really flow in multiple directions. If you put a drop of ink in the water, it will flow everywhere homogeneously. But if you put a drop of water in a sponge, it will follow the thread into the sponge, so it will not be so much anisotropic. And what we found was that there was a problem. The anisotropy was very low with homogeneous tissues at a young age, tended to be higher in middle age, and then went down again. That we couldn't really understand. The first part is easy. There is fat that is building between the myofibrils. They really orient the water in one direction. But what was happening old age was really unclear. So in order to understand that, we had to use a different technology. And we did a very special type of microscopy that allow you to do three-dimensional reconstruction of small tissue. This technology is called FIB-SEM. And in order to do that, I worked with the two incredibly talented microscopists at NCI, which are Lisa Artner and Shiran Subramanian. So this technique, in reality, take out a layer of your tissue, make an image, another layer of the tissue, make another image, and then with a very long process, you reconstruct the three-dimensional images. And I'm just showing you one of them. This is one of the images. This is for an old person. I think this person is 82 years old. And I'm going to show you the movie because you can see the digital images that are reconstructed as a movie. And there are many things that you need to observe here. First of all, this is a capillary. Around the capillary, there is a concentration of subcircular mitochondria. And then you have these sarcomeres that are not very well filled with mitochondria. And then you see this fracture among the myofibrils. The myofibrils have lost their continuity, and that is why diffusion of the water is no longer oriented in a certain direction, must start to be scattered again. And the images become even more evident when you do the three-dimensional reconstruction. So in order to show you better, I have three typical images for young, a middle-aged, and an old person, and I remove the contractile protein. You only see the Z line, which are the line between the different sarcomere, one after the other. And this is a young person. You can see really well in pink the mitochondria, which have a structure that is reticular across the myofibrils very well. And then there's this deposition of this material in small quantity that we don't exactly know what it is yet, but we will. And then when you look at the 54-year-old person, you see, first of all, the myofibrils are no longer perfectly rounded, but they change in shape, and there is disconnection between the different mitochondria that have important functional consequences. If the mitochondria do not create this network, especially at the center of the fiber, you have a deficit in energetics. And then if I show you what's happened, let me see if I can can do that. I want to show you. Well, maybe I can do this and go back and show you what is happening in an old person. See how much the mitochondrial reticular network is disconnected and underrepresented, and there's deposition of proteins that we don't know what their nature, but I suspect strongly that degradation of mitochondria that do not undergo the proper autophagy. Okay, so this first part of my talk really wanted to show you how, if you apply the proper technology, we can gather information on the biological mechanisms underneath, change the curve with aging, and then, with some effort, produce targets. I don't know, do I have still a few minutes? 5 minutes, okay. I'm gonna walk you through quickly the study that I think is gonna open a new can of worms in the longitudinal studies of aging, which is called Gestalt. And I want to say that this could only be done in the intramural program, where there is concentration of incredible expertise all around, but especially we are allowed really to dream. You're allowed to address the basic question instead of having, a very specific hypothesis. And I feel very privileged to be there and be able to do that. So the idea of Gestalt is that we have been working around and around the same problem, the same biomarker for many, many, many years. And I see many times the same paper that is written again and again. And I was kind of tired about it. And so I say, in order to understand more, we need to go back to the drawing board and start doing discovery. And so instead of doing large epidemiological study, we go in a smaller pond, but it's much, much deeper, where we can get, a signal that look without any assumption to the many mechanisms that occur with aging. So what we did is to select 100 people. We haven't finished this study yet. Dispersed over a wide age range. These people are extremely healthy. I'm not going to give you the idea how healthy they are, but the 42 criteria we use are incredibly strict. And then we do cytopheresis in these people. So we collect enormous amount of white blood cells, and through 2 days of 6 people working without sleeping, we get 10 different cell types, and then we do muscle biopsy, skin biopsy, and nasal mucosa biopsies. And in all this material, in single cell, we do all type of omics you can think of. We do genomics, we do metabolomics, gene expression, proteomics, and methylation. So imagine doing 15 samples in each individual every time you do a study. You realize that you can't really do 1,000 people, but you can get enough information for hypothesis generating in 100 people. And this is what we go through. These are the incredible people that are so dedicated to the study. Without, I couldn't do that. I just want to show you one thing. This is a preliminary analysis of the gene expression. And you can see that when you look at the PBMC, which is what everybody does, the signal for aging, it's modest. But when you look at single cell, and especially when you look at muscle, the signal for aging is massive, it's much more evident. So by looking at a mixed bag of cells, you're losing so much information about aging that really we need to find ways to overcome this limitation. And then one of the questions that everybody is asking, there is something that is pervasive, is some signal that is true across every cell types. And there are genes that seem to be upregulated with aging. These are only 25 individuals across 13 different cell types. But look, even 25 individuals, you get a nominal p-value to 10 to the minus 28 with a regression line in each cell type that is absolutely constant. And this ATRX is a very important gene because it's an alternative pathway to the elongation of telomeres. So that makes sense that this is absolutely upregulated with aging. I'm going to skip this. I'm just going to finish with this. These are proteomic analysis of muscle cells. Shotgun proteomics, we were able to look at 4,000 protein in each sample. And here I show you the relationship between shotgun proteomics and self-reported physical activity, something as weak as how much do you are active during the day. And look, 90% of the upregulated protein are mitochondrial proteins. 90% is just something as weak as asking people how physically active they are. And then we look at aging. Of course, we found that many of the downregulated proteins are mitochondrial protein in the muscle. But what is interesting is that not all of them. Only those that belong to the electrotransportation chain, and no structural proteins seem to be downregulated. And so you see that 9 of the 20 proteins downregulated belong to a single complex, which is the ubiquinone complex. So there's something that connects ubiquinone and complex 1 that seems to be downregulated with aging. Okay, I think and you know, what you see is that you know, clearly electrotransportation change and integration of energy metabolism are the two pathways most downregulated. But what is really interesting is that glucose metabolism seems to compensate. So the muscle of older people appear to rely more on glycolysis rather than on oxidative phosphorylation. And we are really currently working to validate and replicate this finding. And again, I to say that, you know, only because I work in a really, really large and fantastic group of people that are so much better than me that I can tell you this story. And thank you very much for your attention.